I am Dr. Nikita Sharma, Associate Professor Chemistry, SPNKS Government PG College, Dausa. Today we will start Unit 2nd Bioorganic Chemistry of M.Sc. Final Year. The first question that comes to our mind where we hear the term bioorganic chemistry is, what is bioorganic chemistry? The term bioorganic chemistry is a combination of organic chemistry and biochemistry. It is that branch of the life science that deals with the study of biological process using chemical methods. Protein and enzyme functions are examples of these processes. Sometimes biochemistry is used interchangeably for bioorganic chemistry. The distinction being that bioorganic chemistry is organic chemistry that is focused on the biological aspects. Biochemistry aims to understanding biological process using chemistry. Bioorganic chemistry attempts to expand organic chemical researches, that is structure, synthesis and kinetics towards biology. Today's topic is enzyme. Enzyme, a substance that acts as a catalyst in living organism, regulating the rate at which chemical reactions proceed without itself being altered in the process. This figure indicates induced fit theory of enzyme substrate binding. In step 1, a substrate approaches the surface of an enzyme in box A, B and C and causes a change in the enzyme shape. The results in the correct alignment of the catalyst groups. Triangles A and B, circles C and D represent substrate binding group on the enzyme that are essential for catalytic activity. Step 2 indicates catalytic group react with the substrate to form products, and step 3 indicates products then separate from the enzyme, freeing to repeat the sequence. Boxes D and E represent examples of molecules that are too large or too small for proper catalytic alignment. Boxes F and G indicates binding of an inhibitor molecule to an allosteric site, thereby preventing interaction of the enzyme with the substrate. Box H indicates binding of an allosteric activator, a non-substrate molecule capable of reacting with the enzyme. The biological process that occurs within all living organisms are chemical reactions, and most are regulated by enzymes. Without an enzyme, many of these reactions would not take place. This includes the digestion of foods in which large nutrient molecules such as protein, carbohydrate and fats are broken down into smaller molecules. The conversion and transformation of chemical energy and the construction of cellular macromolecules from smaller precursors. Many inherited human diseases such as albinism and phenylketonuria result from a deficiency of a particular enzyme. Enzymes also have valuable industrial and medical application. The fermenting of wine, curdling of cheese have been practiced from earliest time. These reactions understood to be the result of the catalytic activity of enzymes. Enzymes have been assumed an increasing importance in industrial processes that involve organic chemical reactions. The use of enzymes in medicine include killing disease, causing microorganisms, promoting wound healing, and diagnosing certain disease. Now, the chemical nature of enzyme. A large protein enzyme molecule is composed of one or more amino acid chains called polypeptide chains. The amino acid sequence determines the characteristic folding patterns of the protein structure, which is essential to enzyme specificity. If the enzyme is subjected to change such for example in temperature or pH, the protein structure may lose its integrity, means denature, and its enzymatic ability. Denaturation is sometime but not always reversible. Bound to some enzyme is an additional chemical component called a cofactor, which is a direct participant in the catalytic event and thus is required for enzymatic activity. A cofactor may be either a coenzyme, an organic molecule such as a vitamin, or an inorganic metal ion. Some enzymes require both. A cofactor may be either tightly or loosely bound to the enzyme. If tightly connected, the cofactor is referred to as a prosthetic group. Now, the nomenclature of enzymes. An enzyme will interact with only one type of substance or group of substances called the substrate to catalyze a certain kind of reaction. Because of this specificity, enzymes often have been named by adding the suffix "ase." Not all enzymes have been named in this manner. However, to ease the confusion surrounding enzyme nomenclature, a classification system has been developed based on the type of reaction in the enzyme catalysis. There are 6 principal categories and their reactions. First is Oxido Reductase. Oxido Reductase means which are involved in electron transfer. Second is transferase. Transferase means which transfer a chemical group from one substance to another. Third is hydrolase, means hydrolysis, which cleave the substrate by uptake of a water molecule. Fourth is lyase, which form double bond by adding or removing a chemical group. Fifth is isomerase. Isomerase means which transfer a group within a molecule to form an isomer. And sixth or last is ligase or synthetase, which couple the formation of various chemical bonds to break down of a pyrophosphate bond in adenosine triphosphate or a similar nucleotide.